Looking at the pictures of these extinct prehistoric animals, it is difficult to believe that they had once walked on our planet. In fact, the Earth and its numerous life forms were originally different. To understand the changes in life forms on Earth over millions of years, we need to know how life originated. The origin of life forms, or biodiversity, is interlinked with the evolution of the Earth, which is again closely associated with the evolution of the universe. The universe is vast, comprises many galaxies, and is almost 20 billion years old. The Big Bang Theory attempts to explain the origin of the universe. According to this theory, a singular huge explosion caused the infinitesimally small, hot and dense universe to expand, which resulted in the lowering of temperatures. It even states that the universe continues to expand even today. After the passage of time, gases such as hydrogen and helium got formed. They condensed due to gravitation and gave rise to different galaxies in the universe. One of the galaxies known to us is the Milky Way, which contains the solar system, and the Earth is one of the planets in this system. At the time of its formation, there was no atmosphere on the Earth. Its surface was covered by water vapor, methane, carbon dioxide, and ammonia released from the molten mass. Gradually, the ultraviolet rays of the sun broke up the water, which was in vapor state, into hydrogen and oxygen. Hydrogen, being a lighter gas than oxygen, escaped. Thereafter, oxygen combined with ammonia and methane, which led to the formation of water, carbon dioxide, and other gases. The high-energy ultraviolet rays of the sun struck the oxygen molecule and split it into oxygen atoms, which in turn reacted with other oxygen molecules to form ozone, and thus the ozone layer was formed. Some of the water vapor cooled and came down as rain, which filled all the deep depressions on the Earth and formed oceans. It is estimated that life appeared on Earth 500 million years after its formation. There are different theories that try to explain the beginning of life on Earth. Some of them are panspermia, spontaneous generation, biogenesis, and chemical evolution. Panspermia is the oldest theory founded by Greek thinkers. It explains that units of life called spores were transferred to different planets. Panspermia assumes that seeds of life exist all over the universe. Therefore, the life on Earth may have originated after one such seed was deposited on the Earth from somewhere in the universe. This theory generates much interest even today. Another belief was that life arose from inanimate things. The theory of spontaneous generation assumed that life came out of non-living, decaying, and rotting matter such as stones, straw, and mud. Even famous Greek thinker and philosopher Aristotle believed in this theory. In fact, this theory was used to explain even Anton von Leeuwenhoek's findings of animalcules using the simple microscope. Moreover, some scientists, such as John Needham, supported the theory of spontaneous generation by conducting some experiments. In one of his experiments, Needham boiled chicken broth in a flask and then cooled it to room temperature in an open container. Later, he sealed this container. After a few days, he observed a few microbes in it. Needham believed that some life force had brought about this spontaneous generation in the chicken broth. The theory of spontaneous generation was in vogue until a German biologist, Rudolf Virchow, challenged it with his theory called omnis cellula acellula, which expounded that living cells can arise from pre-existing cells. However, he could not prove this theory of biogenesis. Later on, Louis Pasteur, with his experiments, proved the theory of biogenesis and forever silenced the advocates of spontaneous generation. In his experiment, he used pre-sterilized sealed flasks with killed yeast. These flasks did not show any signs of life. On the contrary, in another flask, he exposed killed yeast to air. Surprisingly, living organisms were found in this flask. Another theory, called the theory of chemical evolution, was expounded by Russian scientist Alexander Ivanovich Oparin and British scientist John Burdon Sanderson Haldane. They proposed that the first form of life originated from pre-existing non-living organic molecules such as RNA and protein. They also pointed out that the birth of life was preceded by chemical evolution, or the formation of diverse organic molecules from inorganic constituents. When the Earth was created, the conditions were high temperature, volcanic storms, and reducing atmosphere consisting of methane and ammonia. In 1953, American scientist Stanley Lloyd Miller recreated these conditions in a laboratory. He created an electric discharge in a closed flask containing methane, ammonia, hydrogen, and water vapor at 800 degrees centigrade and observed the formation of amino acids. Later on, while conducting similar experiments, other scientists observed the formation of sugars, nitrogen bases, pigment, and fats as well. Interestingly, when meteorite content was analyzed, similar compounds were found in them, which proved that similar processes occurred in other parts of the universe as well. Thereafter, the theory of chemical evolution was most widely accepted.
What is chemical evolution? Scientists have reason to think that the first living cells on Earth came about through a natural process called chemical evolution. Chemical evolution refers to changes in things that need not be capable of reproduction. Examples could be individual molecules or entire chemical systems. A chemical system is a group of molecules that interact with each other. Molecules, structures, and chemical systems almost always evolve or change over time, but they often evolve towards simplicity. Solid iron corrodes into rust when it comes in contact with water. Proteins break down when exposed to too much heat. If simple chemistry is to give rise to something advanced enough to reproduce, there must be situations in which chemical systems can grow in complexity, form new structures, and gain new functions. In order for this to happen, reproduction, which is needed in biological evolution, can be replaced with a much simpler process: repetitive production. On planet Earth and all throughout the universe, powerful natural events take place in regular cycles. The heating and then the cooling of day and night, the repetitive eruptions of volcanic geysers, the rise and then the fall of ocean tides. These events repetitively produce or give birth to new molecules and chemical systems. These products increase over time and often develop new abilities as they interact with their environment. Let's see an example of how this process works. This here is a special molecule called a fatty acid. It's a collection of carbon atoms, hydrogen atoms, and oxygen atoms all stuck together in a specific pattern. Fatty acids are one of many complex molecules that living cells use inside their bodies. They build fatty acids with atoms they get from their environment. Scientists used to think that living cells were the only things able to consistently build fatty acids, but check this out. Lab experiments have shown that if simple common gases, carbon monoxide and hydrogen, are heated up with minerals like those found in the Earth's crust, a variety of complex carbon molecules, including fatty acids, begin to grow. Living cells are not needed. This can happen naturally in underground chambers, heated by the Earth's magma. As pressure builds, these molecules can belch up into pools of water, where a simplified version of natural selection then takes over. Most particles blasted into water will either float or they will sink. Nature selects against them staying in the watery environment. Fatty acids, however, remain suspended in warm water, growing in number as the cycle repeats. When fatty acid concentrations are high enough, they bunch together, automatically self-assembling into a stable ball. This happens because water molecules are attracted to the oxygen heads of fatty acids, sort of like a magnet, but water repels their oily carbon tails. When fatty acids pass near each other, their tails are pushed together by water, eventually forming a ball. As fatty acid collections continue to increase, they join together to make large skins. If fluctuations in these skins happen to make the edges touch, water forces those edges to fuse together. The end result is a stable hollow container, similar to the membrane or skin of a living cell. These containers have a brand new ability. They can trap other molecules inside, acting as an entirely new environment for chemical evolution to continue working within. it's important to note that these membranes do not qualify as living creatures. They can't reproduce on their own the same way living cells do. That said, the development of these membranes, along with many other molecules and chemical systems that scientists have observed, demonstrate an extremely important principle. Chemical evolution can give rise to new characteristics and abilities. Because of this, scientists hypothesize that under the right circumstances, chemical evolution could give rise to systems that are fully capable of reproduction. If they are correct, this would bridge the gap between chemical evolution and biological evolution, demonstrating that chemistry can indeed give rise to life. Scientists at the Center for Chemical Evolution and other research groups around the globe are working hard to test this hypothesis. What is the RNA world hypothesis? If you were to go back in time 120 million years, you'd find yourself in a dinosaur world. 500 million years ago was a world of trilobites and other strange sea creatures. 3.4 billion years ago was the world of the first living cells. And if you were to go back further still, scientists suspect that chains of a chemical called RNA, or something similar to RNA, kickstarted this entire beautiful mess that we call life. RNA is thought to have given rise to life for several reasons. Chains of RNA are found abundantly in all living cells today. RNA is a close chemical cousin to DNA. And with very little help from researchers, RNA chains can replicate, evolve, and interact with their environments. While many details have yet to be worked out, the RNA world hypothesis is the simple idea that somewhere on our early planet, perhaps in a tide pool or hot spring, the Earth's chemistry was producing random chains of RNA. Once formed, they began replicating, evolving, and competing with each other for survival. As these chains evolved and diversified, some eventually began cooperating to produce the genetic code, a wide array of complex proteins, and even living cells which, from the perspective of RNA, can actually be thought of as houses or survival machines for RNA to live inside. To understand how RNA chains can interact with their environments, replicate, and evolve, we first need to understand the simple process of base pairing. Chains of RNA are made of nucleotides, small molecules that come in 4 different types labeled A, C, U, and G. The backbone atoms of a nucleotide, shown here as a yellow bar, can form strong chemical bonds with the backbone atoms of any other RNA nucleotide. This means that different chains can have completely different sequences from left to right. The parts we call the bases of nucleotides, the colored sections labeled A, C, U, or G, are attracted to other bases sort of like a magnet, but they're selective about who they will stick to. G selectively pairs with C, A selectively pairs with U. When bases find their matches and stick together, we call it base pairing. Researchers have found that with a little bit of assistance, base pairing allows chains of RNA to replicate and evolve. Here's how it works. When a long chain of RNA is suspended in cool water with high concentrations of free nucleotides, the chain can act as a template for its own replication. Nucleotides automatically base pair with their partners on the existing chain. If their backbone atoms form chemical bonds with each other, and by the way, this is the part that currently requires assistance from researchers, we're not yet sure how this would have happened in the wild, a complementary RNA strand is born. One with the exact inverse sequence of the original. If the water is then heated, paired bases lose their grip, allowing both chains to act as templates when the cycle repeats. The great thing about this process is that every other RNA chain produced is a copy of the original, but sometimes mutations slip in. This means that as chains compete for survival and reproduction, true evolution, descent with modification, acted upon by selection, can operate on chains of RNA. As amazing as replication is, base pairing also gives RNA chains a second special ability. When placed in water cool enough for base pairing, but without enough free nucleotides for replication, chains will fold up and base pair with themselves. The end result is a complex shape with certain sticky bases pointing outward because they weren't able to find partners. These sticky outward-facing bases can cause unique chemical reactions by interacting with other molecules in their environment. A folded chain of RNA capable of guiding a specific chemical reaction is what we call a ribozyme. Some ribozymes break certain molecules apart, others join certain molecules together. A ribozyme's specific function is determined by its specific shape, and its shape is determined by its sequence. If a mutation changes a ribozyme's sequence, the shape can be modified, and so can its function. When ribozymes were first discovered, scientists wondered how difficult it would be for random chains of RNA to evolve legitimate survival functions. Imagine, for example, a ribozyme that could build nucleotides out of molecules it finds in its environment. Across multiple generations, natural selection could promote and refine this ribozyme because the chain would tend to have access to more free nucleotides than its rivals, allowing it to replicate more often. To explore this idea, researchers at Simon Fraser University produced a large group of random RNA chains and examined them to see if any happened to be able to make nucleotides. Surprisingly, some actually could, but they weren't very efficient. Researchers selected out the successful chains and then used a lab technique called PCR to quickly replicate them with slight random mutations. After just 10 rounds of PCR, followed by selection, highly efficient nucleotide-building ribozymes evolved. These are molecules with the lifelike ability to actively participate in their own survival. These ribozymes, and many others produced through similar experiments, are beginning to blur the line between living things and simple chemistry. So to sum things up, the RNA world hypothesis is the simple idea that the first things to replicate and evolve on our planet may have been chains of RNA or something similar to them. While the basic idea of the RNA world does seem to give us a promising pathway to the origin of life, it's still very much a work in progress. As mentioned, one of several unsolved problems is, how did nature get backbone binding to function without the special enzymes or lab techniques we use today? While many researchers continue to focus on RNA, others are investigating alternative molecules, chemical systems that might replicate and evolve without assistance and could have given rise to RNA. Continual breakthroughs are being found in both avenues of research. I'm John Perry, and that's the RNA world hypothesis, stated clearly.